Hi everyone, I just want to confirm that you can see the slideshow that I have up, right?
Yes.
Perfect. Thank you so much. So we're going to go ahead and get started. It is so exciting to see you all here. Good evening, everyone, and thank you for joining us. Before we get started, I would like to clarify that we are operating on a high school budget and therefore using the free version of Zoom. If the event runs past Zoom's 40-minute limit, I will end the meeting and restart, or send you all an email with a new link if need be. This event has been organized by North Broward School's Sigma Xi Scientific Research Club. Our club was founded in 2021 and granted Explorer Club status earlier this year by Dr. Richard Watkins, Sigma Xi's Director of Membership. Since then, we have published the first edition of Diffusion, our school's new scientific research magazine. We have also held fun events to teach students how exciting science can be, like a pumpkin explosion fundraiser. Perhaps most importantly, though, we have cultivated a love for science in our environment, engaging with publications like Science News and local universities, and mentoring club members as they compete and complete with science fair projects. This evening, you will hear from 3 researchers, each experts in their fields. Our first presentation will be by Ms. Lisbeth Amador, a PhD candidate at the University of Maine. She was formerly affiliated with the University of California, Monterey Bay. Today, she will be speaking about coastal ecology with a presentation called Grazed and Unfazed, Intertidal Urchin Herbivory. Please welcome Ms. Amador.
Hello, everyone. And then Sarah, the screen sharing has been disabled on this one.
Thank you for letting me know. That
Yeah,
should
no
be
worries. Okay. All right, can everyone see my screen all right? Sweet. Okay. Hi, everyone. I'm Liz. As mentioned before, I am a current PhD candidate in ecology and environmental science. But today I'm going to be talking to you about my previous and ongoing research affiliation with Dr. Allison Haupt and her graduate student, Isaac Happerman, on intertidal urchins. And so I'm going to share my pathway to start. I think it's helpful to learn about different career paths and how people got to where they are, as they're not always linear and they can be pretty different. So I grew up in Orange County in Southern California. It's extremely dense, and just to put into perspective, half of the state's population lives in just this southern region. And so I moved to Central California for my undergraduate at Cal State Monterey Bay, where I studied marine science. I took a year during that to study abroad in Australia, where I had the amazing opportunity to volunteer in a seagrass restoration program. And came back, and after 5 years, I graduated with a major in marine science and a minor in statistics. I took a 6-month gap after as a research scholar for an NSF-funded GeoBridge program, where I modeled urchin populations. And I learned that I liked research and ecological modeling. I was encouraged to apply for PhD programs, where I landed a position here at the University of Maine, where I am modeling time sensitivities to climate change and land use. But I will be focusing on my intertidal urchin research with Allison and Isaac. And so, for those who may not know, what is the rocky intertidal? Well, the intertidal is the seashore area that is exposed at low tide and covered at high tide. It's dominated with rocks that are large or small, but not sand. One way to think about it are tide pools. And along California, it has a predominantly rocky coastline. So it's a bit different to the sandy coastlines along Florida. And these are temperate oceans. So it's the middle between the really cold polar waters and tropical warm waters. And so for those attending today, just go ahead and shout out or in the chat what you might think you'll find in a rocky intertidal. Right, yep. Yeah, so among some things you'll find algae, crabs, there's definitely fish, your stars, clams, and mussels, fun worms, your sea urchins, and colorful worms. And so many other things as well. And so the Coastal Ecology Lab, run by Dr. Allison Hout at CSUMB, go across different sites along Monterey Bay, California, and conduct these intertidal surveys, which is also a predominantly rocky coastline. So I focus on intertidal purple sea urchins. As you may have gathered, they are algal-eating invertebrates that only live in these rocky reefs, and they mainly sit around and wait for their food to drift on by. They're like these little couch potatoes hiding within crevices and reefs, and they just clutch onto their food. But when conditions shift, they will emerge from these places and actively graze on algae. They're not toxic, but their spines can hurt, can do some damage, and they can reach a diameter of up to 2 inches, like the size of a little fidget spinner. And something you can do with urchins is count them to get a general idea of how many are present within an area. And so we go across these sites and conduct intertidal urchin surveys at low tide, use observational techniques that can be done over long periods of time. We lay down these transit lines, which are essentially these long, flimsy rulers. We count urchins that are about nickel size or greater along these transects. We also lay down these quadrats or squared grids and take note of the total area covered for different types of algae. And as a disclaimer, the Hop Lab does not have an official TikTok, but maybe in the future. This is just a video to show us out in action, what the field sites look like, and most importantly, us having fun while doing it too. Okay, and so another tool that can be used for intertidal surveys are drones. And this is done, we did this for the lower intertidal since it's the more hazardous area and we can't safely survey there. And it's a recent thing I started within the lab. I did not fly the drones, but that's something you can definitely do if you're interested in, get a certification. I managed and coordinated the flights with the tech officer and drone pilot, Pat and Pietro, using drones from the Marine Landscape Ecology Lab at Cal State Monterey Bay. And so in the top left, this is kind of the perspective you'll see when you're at the site. And then you'll go ahead and fly the drone. And for those who don't know, you control them with the remote. And so this is the little interface. And then this is the image that you can get from the drone. And you can train an algorithm to detect the seagrass, algae, whatever you're looking for, and conduct analyses with it. So coming back to these urchin data that we collect, a way to visualize this is to graph it. And so we take these urchin counts and calculate densities or the number of urchins within an area. And then we can plot it across time as well. And so another question for chat. What can anyone kind of get an idea of what the trend is or how urchins are changing across the years?
Well, we could clearly see the urchin population doubling amongst the years to 2020 to 2022, 2021, sorry.
Yeah. Yeah, so they're doubling, they're increasing over the years. That's great. Our data shows that they've starting to increase from 2019 to 2020, and at these high rates as well as you've mentioned. Which is wild.
Yeah. Do you know why this happens? So maybe what's the reason for that, such a drastic increase in local population?
That's a great question. So that's part of the research. 7, 8 years ago, it would have been difficult to find them in the intertidal. They would have been within their crevices, but now we're seeing them sprout out across the rocks at low tide. And this is kind of a relatively newer phenomenon for the intertidal. And so that's what we're trying to figure out. That's a great question. But yeah, so these are like before and after pictures of these urchin populations. And part of my research is trying to find out where do we find these intertidal urchins and why, and what their impacts on intertidal communities are, using this observational and field data as opposed to like experimental data. Some tools we can use to answer these questions like Excel or some coding software programs, much more. But essentially, we use these to do statistical analyses to see how meaningful these relationships or differences between the things of interest are, in our case, urchins. And so some questions we've asked are intertidal urchins different across space or across sites? And if they are, what's driving? So why is there this increase in urchins, and why are they different across space? And then what are the impacts to their surrounding communities? And so one way we've approached the first problem is to kind of look at the densities for this. And so circles are urchin densities, and the size of circles is how dense it is. And so chat again, are urchin populations different across space? Can you see some kind of difference?
We can clearly see that at the Catania Pescadero point, there's a very large kind of a bubble of them. If not China Rock and Point Pines, there's like not really a lot of them.
Yeah, so there's so much more Pescadero Point that we found compared to the sites with smaller circles. And that's what we found as well, that they're patchy across these sites and they're different, despite being at these high densities, as we saw in the previous graph. And that's how we kind of came about our first question. And so for our second question, we're now looking at the characteristics within the ecosystem to kind of determine that why are they increasing and what's influencing these differences in densities. And so this is kind of like a beefy way to look at it, all these different interactions, different factors between each other and with these intertidal urchins. And so it can be a little complicated to look at. And so here's a more fun and kind of simpler way to look at it, where we have our physical factors of wave energy and habitat complexity, and then food factors where we incorporate fleshy algae from different intertidal zones, so the lower or the middle, and then interactions with neighboring populations that's in a different type of habitat. And so we're all looking at how they interact with each other and with intertidal urchins as well. And this is one of the things I'm currently working on. And in addition to this, which is part of my collaboration, we're looking at community impacts, so that third question. And we're going to be using algae and invertebrate data, so looking at mussels, limpets, anemone, and other critters to see how this has changed before and after the increase of intertidal urchins. And so it's part of that ongoing why they're increasing and kind of what's kind of driving these differences in densities across these sites, and then those implications to the community. So to kind of tie this all together, yeah, you may be wondering, why are these urchins increasing, and is this a bad thing? We do not know that yet for sure in the intertidal. This increase is a relatively new thing for the intertidal. But what we do know is that they can have negative implications to the biodiversity and functionality of the ecosystem. And we know this from kind of increasing populations within that other habitat. That's kind of really left a detrimental blow to the ecosystems there. And another thing is ecological monitoring is important because you can inform and create proactive management strategies. And then in addition to that, it's important to look at these different lens of research. So looking at different avenues within the field, where we're looking at this small population lens, where we're looking at urchin population change, and at this larger lens where we're looking at these overall community change and interactions. And so this is kind of what I had planned for my talk, and I'd like to thank the intertidal field team and volunteers for coming out and covering hundreds of urchins. As you can see from the graph, there are a lot. Pat and Pietro, drone pilot, the Marine Landscape Ecology Lab for providing drones, and our funders. And then I provided my contact information in case anyone would like to reach out and learn a bit more about coastal ecology research and how to get involved. And so yeah, and I'll be taking any follow-up questions, and I guess this starts our Q&A for my section.
If I would like to start off, so I've listened to your presentation. Well, first of all, very thank you for your time and coming to us and to actually talk to us. So my question is, might the population of the orchids be impacted by there's an absence of natural predators in the area? Can this be the case that we see the sudden increase in the population?
Right, so just to rephrase your question, like why is there an increase despite having predators in the area?
Yes.
Yeah, so kind of part of these increases has been like the combination of so many different things that happened at once. One of their main predators have been like the ochre sea stars, sunflower sea stars, excuse me, which have been detrimentally hit with a disease that essentially causes these sea stars to melt. And so there is kind of this like decrease in predators for urchins. But yeah, that's one of the things we kind of haven't we're planning to take into account, is to really look at that predator aspect, because there's still so many of them and they still seem to still be increasing despite having other predators like birds, seabirds in the area too. So hope that answered your question.
Okay, thanks a lot.
Yeah. There's a question in the chat. Can these sea urchins be found in any places other than intertidal? Yes. These other habitat type that they can be found is within the subtidal. So this is the area just adjacent to the intertidal, and this is the rocky reefs that are constantly being submerged underwater. And in these places, you'll find larger algae. So you have these like big kelp forests, which they also love to eat. And this is part of why we know they can have negative impacts, because their populations in the subtidal have increased, and they've essentially can mow down these kelp forests and these other algae types, leaving behind these like barrens that are unproductive. But yeah, the subtitles and other places you can find these urchins.
How long did you study it for being this? So how long did it take for you from school to go to university and stuff like that?
Like to study urchins or?
Well, how long did it get to you? One second. So how many years did it pass from the university to actually applying as a PhD candidate?
Oh, okay, yeah. So only 6 months. After graduating from my undergrad, I was doing this kind of research with urchins, and then a little closer to like 3 months into my program, I started looking for graduate schools, and that was about a 2 to 3 month process for me. So I was only out of school for half a year before I jumped back into graduate school. And it's different for everyone. Some people take like 3-year gaps or longer, or they go straight into graduate school. So it all depends on where life takes you. And there's some more questions in the chat. Are there differences among species population size, or is there only one species? Ooh, yes. So there is another type of urchin, which is the red urchin, and those can get a lot bigger. Those population sizes are different. I think I've only seen 2 red urchins while out in the intertidal, counting a bunch of purple urchins. So yeah, there's a difference between that too, which is interesting to see these differences in species populations. And another question, where do you think it would be beneficial to survey in the future? So can I get a little bit more context on the where, Emily? Like, do you mean more sites or in different habitats? Oh, yeah. So I think it would be beneficial. Part of it is kind of urchins. You can find purple urchins down from Baja California up to Alaska. And there are long-term ecological research organizations like PISCO and Marine in these areas that do general intertidal surveys, but they also look at purple urchins as well. And in terms of our study, it would be beneficial to expand. That way we get more of an idea of a bit more variety within these sites. It's not different. Monterey Bay is a bay, and then you have this like the outside of the bay, which experiences different conditions. And there's tons of surveys being done in the subtitles. So subtitle urchins are a more well-studied population compared to the intertidal. Yeah. Any other questions? I think I'm coming to my time for the Q&A.
Perfect. Thank you so much for that amazing presentation. I don't know about you all, but I especially loved the Spider-Man meme that explained causes for urchin population differences. Our second speaker today was unable to make it, so today's final speaker is Mr. Akash Nathani, a PhD student at Florida Agricultural and Mechanical University. He has been affiliated with organizations like India's National Institute of Pharmaceutical Education and Research and the Holy Mary Institute of Technology and Science. His presentation today is titled Natural Killer Cell-Derived Extracellular Vesicles for Lung Cancer Immunotherapy. Please welcome Mr. Nathani.
Hi, everybody. As Sarah said, my name is Akash, and I'm a PhD student for A&M University. I am also the president of my university's American Association of Pharmaceutical Scientists student chapter, and just wanted to ask a few questions before I move on to my topic. So can anybody tell me what are the three leading causes of death? Any guesses? Top three leading causes of death.
Probably heart attacks, strokes, and car
Yes,
accidents.
that's true. First one is heart problems. Second one is, what's the second and third?
Strokes, like brain strokes.
No, it's not. Second one is cancer. Cancer, yes. Third one is accidents. So, so heart problems and accidents, they're not under my control. But what I am doing in my research is working on the top two problems, which is heart problems and cancer. And what got me to working in cancer is, I had few personal incidents where I lost a couple of my dear ones to cancer. And then my love for biology, chemistry, physics, all the branches of science brought me to pursue bachelor's in pharmacy. Pharmacy has everything: biology, chemistry, physics. And then I went on to pursue masters for doing more research on cancer. But then COVID hit. I was not able to do a lot of things in the lab. I was there only for 6 months in master's degree. Then I went on to do. Then I thought of doing PhD in Florida A&M. That's where I am now in my second year, and I am working on I'm working on treating lung cancer. Lung cancer is the second most and common in both males and females. Whereas breast cancer is the leading cause of death in females and prostate in males, but lung cancer is affecting both equally. So yeah, but the problem with treating cancer is that there are treatment options. There are radiation therapies. There are surgical therapies. There is chemotherapy. But surgical therapy, or be it radiation, there's a lapse again. There's time waste. It's expensive. And the most common treatment is chemotherapy, where you use drugs. You use these chemical drugs, but these drugs have side effects. The most common side effect, people don't like it, alopecia, which is hair loss. When you use these drugs, you have hair loss. People's confidence go down. People don't like it, and then there is fatigue, diarrhea, constipation, all kinds of problems, infections, inflammation, pain, everything. So how do you overcome this problem of existing treatments? There's something called immunotherapy. There's when you have infections, right? Your body's body's immune system has a mechanism to fight against it. But when it comes to cancer, your body is not able to fight against the cancer cells. So these immune cells are deprived of their own function. So what I am trying to do is somehow enhance the immune response in the body. Not treat the cancer cells by myself, but help immune cells treat their own cancer body, right? So that will be my topic, and let me share my screen. Can you see it? Can you see my screen?
We can, yes.
Yes,
Okay,
yes.
perfect. Okay. So what I'm doing is trying to treat lung cancer using immune therapy, not chemotherapy, not other kinds of therapy, using something called EVs. These are not electric vehicles, but these are extracellular vesicles that are secreted by every cell in the body. Every cell in the body secretes EVs, which are extracellular vesicles. But what I'm trying to see is if these EVs have any anti-cancer properties. And after a literature survey, I found out that if you get these EVs from immune cells like mast cells, T cells, B cells, dendritic cells, natural killer cells. So all these EVs have anti-cancer property because natural killer, for example, natural killer cells. So these natural killer cells have cytotoxic proteins in them, which are secreted into their EVs. So all these cells, natural killer cells, which release EVs, have they're acting as parents, and these EVs, which are children, also have the same properties. So instead of using a tablet or a capsule or syrup or a solution with drug inside it, I'm trying to see if these EVs can be used as a drug or medicine. So before going to that, let me talk about what I do, where I did my master's. I did in my bachelor's and master's in India. Then my research interests are cancer specifically. We also work in neuropathic pain. We also work in heart problems. But my own research area interest is cancer using exosomes. Exosomes are nothing but the EVs that I was talking about, and how to deliver a drug and solid state pharmaceutics. So the purpose of this session is to explain you how I can use these EVs to treat cancer with the term called immunotherapy. And then I perform few tests to see if that works. And then first I have to isolate these EVs from the cells. So once the cells release these EVs, I have to isolate them, then characterize them if they are EVs or not. And then, if I want to combine both chemotherapy and immunotherapy, because I believe chemotherapy or immunotherapy alone would not be sufficient. So we may have to combine both of them, or maybe even when you're combining chemotherapy to immunotherapy, uh, there are advantages and disadvantages of both the therapies. But when you combine them, the advantages are there, but the disadvantages go away. Let me take an example. If you take a drug alone in chemotherapy, and if you're combining immunotherapy, you can reduce the dose of the drug, which can reduce the side effects. So that's how you can overcome these disadvantages of using chemotherapy. So what are these EVs? What are these? So they are lipid bilayer particles ranging in the size of 40 to 5,000 nanometers. Based on the size, they are called with different names. 50 to 5,000, they are called apoptotic bodies. 50 to 1,000, they are called microvesicles. And the smallest one, 40 to 100 nanometers, are called exosomes. But they are all EVs. My main focus is on exosomes because of the size. If the vesicles are smaller, they'll be able to easily penetrate the solid tumors which I'm targeting. So that's why I'm focusing mostly on EVs. The exosome EVs, which are around 100 nanometers size. Why am I choosing EVs over other systems like capsules and injections or syrups or solutions? Is when you give a drug from outside, your body thinks that this is a foreign particle and acts against it. So this EV is from your own body. I'm taking cells which are from human cells, right? So I'm taking these human cells and getting EVs from them. And when you give EVs to the body, the body does not consider it as a foreign particle. So it avoids phagocytosis, which is cell eating, which is cell killing, cell death. So that doesn't happen. And when you give a drug inside the body, it has a limited period of circulation. The half-life is very less. It is getting excreted when you urinate or via sweat, via urine, via feces. It gets out of the body very quickly. So you have to keep on administer the drugs every day. But not it's not the same case with EVs. And these EVs also have a ability to cross physiological barriers. The most difficult barrier to cross is blood-brain barrier. If you want to have an effect on brain, like anything which is related to let's say depression, Alzheimer's, or Parkinson's disease, you cannot treat them using conventional therapies. You have to use something which is very small, that can penetrate these barriers. So EVs can also do that, and they are naturally stable, and they have a nice cytotoxic potential. That's what I'm figuring out now. So why, out of all the cells in the body, why natural killer cells? Why do I have to choose natural killer cells as source of my EVs? The choice of my cells was very important. I cannot choose any other random cell in the body and expect those EVs to have anti-cancer property. So why did I choose natural killer cells? Is because natural killer cells are known to have immune response and are known to have anti-tumor properties. But I hypothesize that these EVs that I get from NK cells also have anti-tumor properties, because they'll have the same cargo, or they'll have the same content inside them. That's what the hypothesis is. And there is enough evidence to show that these exosomes or EVs have inhibited tumor growth for example, in neuroblastoma. And I'll just skip this. And why do I think that? Is because there are certain proteins in NK cells which are called perforins and granzymes, right? So these perforins and granzymes are very well known to have anti-cancer properties. And if I can prove that EVs also have these proteins, which is perforin and granzyme, they'll also be able to kill the cancer cells. So how did I come to know that a natural killer cell derived exosomes can work? After doing a thorough literature survey, I found that natural killer cell derived exosomes can have enhanced anti-tumor effect. They can also be used in neuroblastoma, which is brain cancer. They can also be used in melanoma, which is skin cancer, right? But how do I get these EVs? They are not sold by any company. We do not have them readily available. So what I do is get the cells, and once I have the cells, after a process of chemical reaction between the EVs and pegylation, and I go with a lot of processes. There is a long protocol for a week or 2 weeks, where you do centrifugation, ultracentrifugation, and then finally characterize them if they are EVs or not. Not talking about it in detail, but this is how it works.
Salt and straw. All the ice cream went on Fillmore, which is close to his house, is hiring.
So yeah, this is how it works. What I do is I get the cells, I grow them in the flask, and then put them in the tube. I spin it down, or at a speed of 3,300 G, I spin it, and then for 10 minutes, I'm able to take out the larger vesicles, which are the cells. Another 10 minutes at a higher speed, I'm able to take out the smaller cells, which are the green ones, or which are the dead cells. And finally, I'm able to do the removal of cell debris at a higher speed. Finally, the red vesicles that you see are the exosomes that I'm interested in. So for that, I do ultracentrifugation at a higher speed, with a very high speed, to get the exosomes that I am desiring. Then I characterize them if they are EVs or not, right? So what I do is nanoparticle tracking analysis to understand if the particle size is around 100 or not, and then determine the concentration and zeta potential to know if they are stable or not at room temperature. And then I do various assays and protein estimation to see if the proteins are present or not, if the proteins like perforin and granzyme, which I'm desiring, are there or not. And that I come to know or confirm by Western blotting and proteomic analysis, which gives me confirmation that these proteins exist in the EVs. Using nano-tracking analysis, I get the size. And using in vitro studies and in vivo studies, which are the cell line studies and animal studies, I confirm that the EVs have anti-cancer properties. What I did is I had 4 groups where the first group is control, where I did not do any treatment. The second one is just NKEVs. The third one is just carboplatin, which is a very well-known standard drug in chemotherapy for lung cancer. And then I combined immunotherapy and chemotherapy, which is NKEVs plus carboplatin, and treated the cancer in mice. I'll show you the results. And before that, I would like you to see that the graph clearly says that these EVs are exosomes, which are around 100 nanometers in size, and the zeta potential of 21 says they are stable. And I had to do BCA assay to quantify how much protein is there in them. Then I compared these EVs with any other EVs. I compared them with MSC EVs, which are mesenchymal stem cell EVs. And mesenchymal stem cell EVs did not have any killing. The cells were viable all the time. No matter what concentration of EVs I use, their cells were viable, the cells were alive. But with NK cells, which are shown in pink, the concentration, there was a concentration dependent cell viability. That means the NKEVs are killing the cancer cells. That was confirmed by Western blotting. And I did proteomics to understand if the proteins are there or not. I was looking for perforin and I was looking for granzyme. Both of them were present in the EVs, which confirm that NKEVs have anti-cancer properties. And I tested them in animals. And animal data, as you can see the graph, x-axis I have number of days, y-axis I have the tumor volume. I treated these animals with NKEVs, then carboplatin alone, and also with their combinations. The combination works the best in a particular mice, and also in athymic mice, which are immunosuppressant mice. Even then, the combination worked the best. The tumor volume was very less, not more than 1000 millimeter cube, even after 10 days, whereas the other treatments had a little higher tumor volume. So I conclude that I was successfully able to isolate EVs and characterize them, and then do a lot of studies in vitro and animal studies, and to tell that these EVs, you may not have heard about them until today, and you may not hear it for another decade or two, but after a couple of decades, EVs will be in the market and will be used replacing the current chemotherapy drugs for treating any type of cancer, because they do not have any immunogenic response, they do not have toxicity for healthy cells, they do not have any phagocytosis effect, and they are not foreign particles, they are from your own body. So yeah, I think they can easily replace the currently existing drugs. These were the references I used for my studies, a lot of them and a lot more. I have not mentioned all of them. And I would like to acknowledge my supervisor and my lab mates, everybody, postdocs, all my lab mates, my colleagues. And if you have any questions, feel free to ask me now, or reach out to me on my email, phone, LinkedIn, anytime. And thank you, Sarah and team, for giving me this opportunity.
I have a question. So,
Sure.
what do you think about genetic therapy for curing cancer? I've recently just done some reading about that, some papers, and I'm really curious about what you think about it.
Right. So gene therapy is what you're talking about, right? The problem with gene therapy is it's not in general. You cannot give a gene to each and every individual. It has to be personalized. You have to take out genes from a person and then give it back to them, treat that gene in particular, because I'm talking about the mutations here. Cancers generally happen when there are mutations in the genes, right? And then you can fix this gene and give it back to the human, and that can have anti-cancer activity. But the problem with genes is it's still expensive. It has to be personalized. It cannot be general. I cannot take a gene from X person and give it to Y person, and that makes it challenging. And the other part is the genes are not stable in at room temperature. So right, so transportation issues, and then they're not stable for a longer period of time. Whereas EVs or any other drug, they have, for example, chemotherapy drugs, they are stable for 1 or 2 years. You see expiry dates of 1 or 2 years. EVs, we have tested them in the lab. They can be stored for 6 months or at least for 3 months. But genes, that's a problem.
Okay, thanks a lot.
Okay. Any other questions?
I think there's one in the chat.
Oh, let me see. Okay, when doing these ultracentrifugation, why can't you take all the cells, dead cells out together? Okay, that is because of their different sizes. Cells have a different size, dead cells have different size, and debris have different sizes. So if I'm trying to run it at a particular speed and trying to remove all of them together, because of the size differences, I cannot choose this particular speed. Let's say if I'm doing it at 1000 RPM, okay, if I'm doing it 1000 RPM, only cells or dead cells might get removed, and then the smaller cells, which are the debris, will be still there. But if I am going at a little higher, for example, if I'm going at 10,000 RPM, at 10,000 RPM, if I'm trying to remove all of them, I'll still have a lot of debris left, because they form agglomerates, they form aggregates, and then at a higher speed they may sediment at the bottom, right? So I have to do it differential centrifugation. This is called differential centrifugation, not ultracentrifugation. Yeah. Do you have any ideas as to why the error margins were higher in the pink viability bar graph than in the control? What sources of limitations did you face in your experiments? Are you talking about the viability graph that I showed? The pink versus the yellow?
Yeah.
Oh, the error margins were high because when I repeat the experiment using these NK cells, the viability was not similar all the time. Sometimes they were killing 25% of the cells, sometimes they were killing 40% of the cells. So so even if I do n equal to 3, the standard deviation was a little higher. But whereas the other one, MSCEVs, they did not kill at all. So it was always maybe 5% killing maximum. That's it. So that's why the error was less.
Do you know why there were the differences between killing 25 and 40% of the cells, or is that still unknown?
Depends, because I'm giving the same concentration, but the amount of protein that these EVs have got from their parent cells might be different, right? In one treatment, they may have 40 to 50 microgram of protein. In the other one, they may have 60 to 70 microgram, and that might be the possible reason, right?
Thank you.
You're welcome. If you have more questions, anymore?
Yeah, I may have missed this early on, but these EVs, do they have to come from the cancer patient to avoid rejection from the body, or?
Okay, I got your question. So there are two types of EVs. One, you can get it from cancer patients. The other ones, you can just make it in a company or make it outside, not depending on anybody. So if you're trying to get EVs from cancer patients, you have to be careful, because when you have EVs from cancer patients, these EVs will have tumor antigens on them, right? So these antigens will act as vaccines. So if you're trying to make vaccines, I'll prefer EVs from cancer patients. But if I'm not I'm not focusing on vaccine delivery, I'm trying to give it as immunotherapy or trying to increase immune system's response against cancer, then I can just get these EVs from immune cells, which will have these proteins which can kill the cancer cells, and give it to a patient whose immune system is not working well. Let's say if my body's immune system is working fine, take out cells from my body, because the cells are already readily available from the you can get it from a company, and get EVs isolated from them, and give it to the cancer patient who is having immune system problems.
And the body won't reject somebody else's immune cells.
Exactly. No,
Huh,
exactly. No,
okay.
they are so small. They are so small that the immune system does not recognize them at first. Secondly, all EVs are from natural killer cells or any other immune cells which are there in every human. So the cargo or the phenotype is not changing from person to person.
Interesting. Thank you.
Right? Yeah.
Thank you so much for that fascinating presentation. So
Okay. Thank you.
I'm going to share my screen one more time and close up. It is customary for us to end our club meetings with a science-related pun. So, as to not curl the tradition, why do biologists look forward to casual Fridays? Because they're allowed to wear jeans to work. I think I'm hilarious. Please feel free to connect with us on social media at SRC and NBPS. That concludes today's event. Thank you for coming.
Thank you, Sarah.
Thank you.
Thanks a lot.
Thank you, Sarah.
Great pun, by the way.
Yeah, great.